Microscopic features. So, what is microscopic? According to Merriam-Webster Dictionary, microscopic it's about resembling a microscope, especially in perception. When you say perception, it is in your own understanding or kung paano mo mauunawaan yung pag-resembling ng microscope. And invisible or distinguishable without the use of a microscope. So, invisible, yung hindi nakikita or you cannot see. Indistinguishable, yung hindi matukoy or unidentified. Then, very small or fine or precise. So, napakaliit at pino daw yun. And of relating to or conducted with a microscope or microscopy. So, conducted, it is how you manage or supervise with the microscope or microscopy. That is microscopic. So, these are the following questions can take identifying microbes using microscope. First is, are you looking at rods, cocci, or spiral-shaped bacteria? Yung rods ay yung makikita nyo yung picture. Ito siya. Ayan yung rods. And yung cocci naman, ito siya, cocci. And spiral-shaped bacteria, ayan siya. Ayan yung tatlong yun. And the second is, are you looking at bacterial cells with flagella? 3, are you looking at bonding yeast? 4, are you looking at the filamentous fungus with branching hypha? So, these 4 questions will help to determine or identify the microbes when using the microscope. The next, and here is the example of microscopic features. These pictures is about microscopic observation of oil droplets under 63 times magnification. The letter A picture, it's called colloid meal homogenization. And the other one, letter B, and the letter B, it's called HDP microfluidization. And the last, this picture, it's called HPP process. That is the example of microscopic. The end. Immunological identification. What is immunological? So, according to Oxford Dictionary, it is a relating to or involving immunity to infection or toxins. So, ayun daw yung mga infection or toxins na involving sa ating katawan or immunity. Then, according to Cambridge Dictionary, it is relating to the structure and function of the immune system, that part of the body that fights off viruses, bacteria, fungi, and other types of infections. So, kung paano daw labanan ng ating mga katawan or immunity ang mga viruses, bacteria, and so on. That is immunological. So, ELISA stands for Enzyme Linked Immunoassay. This method, it's for microbial detection. That usually within a diagnostic on a species by species basis. Then, An ELISA test may be used to diagnose HIV, which causes AIDS and Lyme disease. When you say AIDS, it is a set of symptoms and illness that develop when an advanced HIV infection has destroyed the immune system. And when you say Lyme disease, it is a bacterial infection can get from the bite of an infected tick. When you say tick, ito yung tinatawag ng mga niknik, pulgas o garapata. Then, the infection can spread to your joints, heart, and of course to your nervous system. So, these methods are highly sensitive, but they rely on very specific antibodies and highly discriminating proteins within the organism of interest. So that's all. Thank you.
Good day, everyone. This is the report of the Group 3. Today, we will discuss the techniques in microbial identification. Identifying microbes at the species level. Species level identification allows you to discriminate between two species from the same genus, which is often essential in the treatment of infectious disease. For example, the bacterial genus Yersinia contains approximately 15 species, some of which form part of the normal human microflora, but others of which are serious pathogens and require medical treatment. Mahalagang i-identify ang isang klase ng bacteria so we can detect these different processes to aid in our identification of unknown bacterial samples. Test results can be applied in medical world when determining an infection and the cause of disease. Staining and microscopy. Stains enable easier visualization under a microscope. Cytology microscopes have specific requirements to ensure clear differentiation between stained cells. Ang purpose ng staining is para ma-visualize yung mga cell under the microscope and to differentiate between live and dead cells in sample. Under the staining, there are different kinds of staining. The first one is gram staining. It's a common technique used to differentiate two large groups of bacteria based on their different cell wall constituents. Gram staining is the most important procedure in microbiology, used to differentiate between gram-positive and gram-negative organisms. Ang purpose ng gram staining is to check for bacteria at the site of a suspected infection or in certain body fluids, such as blood or urine. Crystal violet dye is a purple stain based on the crystal violet dye, is named after the Danish bacteriologist Hans Christian Gram, who developed gram staining. Crystal violet is not only used as a textile dye, but also it is used to dye paper and as a component of navy blue and black inks for printing, ballpoint pens, and inkjet printers. Makikita mo rin daw ang crystal violet sa mga printer, sa mga ballpoint ng ballpen. Gram-positive and gram-negative. Gram-positive bacteria have a thick peptidoglycan layer and no outer lipid membrane. Gram-negative bacteria have a thin peptidoglycan layer and have an outer lipid membrane. Ito ang itsura ng gram-positive. Mas makapal ang peptidoglycan niya kaysa sa gram-negative. Under the microscope, ito naman ang itsura ng gram-positive bacteria at gram-negative bacteria. Endospore staining. This involves applying a stain to a bacterial sample to check for the presence of spores. Because not all bacteria produce spores, this information can be useful in identification. Several spore stains are available, but malachite green is probably the most popular. The purpose of endospore staining is to differentiate bacterial spores from other vegetative cells and to differentiate spore formers from non-spore formers. Endospores are formed by a few genera of bacteria such as Bacillus. By forming spores, bacteria can survive in hostile conditions. Endospores can form within different areas of the vegetative cell. They can be central. Endospores are located with the middle of the vegetative. Terminal endospores are located at the end of the vegetative. Subterminal endospores are located between the middle and the end of the cell. Ziehl-Neelsen staining. This is an important tool for the staining of Mycobacterium tuberculosis, which can be gram-stained. The purpose of Ziehl-Neelsen staining is a bacteriological stain used to identify acid-fast organisms, mainly by Mycobacteria. Ziehl-Neelsen staining was introduced by Paul Ehrlich. It is named for two German doctors who modified the stain, the bacteriologist Franz Ziehl and the pathologist Friedrich Nielsen. Stains for fungi and yeast. Several fungal stains exist, although they are generally non-specific. Fungi and yeast are stained purple to red. Concentration of lactic acid in the stain mixture plays an important role in the differential staining of pollen. For staining fungi yeast, the stain has to be acidic, but its concentration is not critical except for bacteria. Examples of fungal stains include lactophenol cotton blue. This stain contains phenol, which kill the organisms. Cotton blue, which stains the chitin found in fungal cell walls. Periodic acid skiff stain. This stains carbohydrates and other moieties and causes a magenta color in living fungi only. This is used in infection diagnosis. Periodic acid skiff is a staining method used to detect polysaccharides such as glycogen, mucous substances such as glycoproteins, glycolipids tissue. Grocut's metamine silver stain. This colors fungal cell walls brown to black but cannot distinguish between live or dead material. Grocut's metamine silver stain, it is used widely as a screen for fungal organisms. Trypan blue, aniline blue, and chalcofluor white. These stain fungal and plant structures and are used in the study of plant-fungal symbiosis and plant pathology. Trypan blue is a diazo dye that has been widely used to color dead tissues or cells.
So, yun. Some main applications of accurate microbial identification. So, first, ano ba yun? Yan, healthcare. So, accurate and fast identification of bacteria, fungi, and parasites is important for correct and timely disease diagnosis and appropriate treatment. So, yan nga, accurate and fast identification. So, yan, like for example, in our future job, this is very indeed, especially sa medical industry na pwede nating pasukan na may kinalaman, especially sa specialization natin na microbiology. So, kaya nga from the word application, so ibig sabihin, halimbawa, doctor ka na or microbiologist ka na, senior later, so kailangan alam mo yung applicable treatment na kailangan ng pasyente mo. So, kaya naman sa laboratory. Yan, kaya dapat application, so ibig sabihin accurate and fast identification ng bacteria, alam mo kung paano i-identify yung isang bacteria, fungi, and parasites, and etc. So, yan, next. Epidemiology. So, ano ba yun? Epidemiology. So, epidemiology is the study and analysis of the distributions, patterns, and determinant of health and disease condition in defined population. So, yan. Identifying microbes is important for tracking and tracing disease spreads and outbreaks, as well as for identifying isolates. So, yan. Tracking and tracing disease. So, yan. Let's say, for example, na-identify na yan. Na-identify na si COVID-19, yan, si virus. So, dahil nga nag-spread na si virus, kailangan natin lahat na mag-undergo sa ECQ, or Enhancement Community Quarantine. Kasi nga, yan, once na naka-ECQ na tayo, madali na yung tracing and tracking ng isang disease na yan. So, yan. Ang pinakang main problem lang talaga is, halimbawa ng mga contact tracer, is, once na-interview nila yung isang COVID positive, is hindi lahat sinasabi nung COVID positive kung saan siya mismo nanggaling, kung sino-sino yung mga kasalamuhan niya. So, mahirap pa rin dito sa bansa natin na matrack kung sino talaga yung lahat na positive. Siyempre, mabilis talaga yung spreading niya. So, yan nga. Kaya nga, ang best solution doon is, para sa akin lang naman, is magkaroon ng bakuna ang lahat. Ayun na. So, next tayo. Ayan, so next na tayo, si pharmaceutical industry na tayo. So, because microbes are a significant threat to sterility, being able to accurately identify microbes is often a good manufacturing practice or GMP requirement within the pharmaceutical industry. So, what is the meaning of this? So, ayun nga, so involved naman dito is yung mga microbiologists and pharmacists, etc. So, yan, once na may accurate knowledge na sila, so let's say for example na microbes or virus, yun nga, mas ma-identify na nila yung si COVID-19. So, ibig sabihin, alam, or pwede na silang gumawa ng gamot na panlaban din sa virus na yun. Kaya, very in lead sa atin sa yung pharmaceutical industry din. So, next natin is additional scenarios. So, yan. Additional scenarios. Criminal investigations, microbial forensics, the investigation of bioterrorism threats, and environmental studies all need to accurately identify microbes for various purposes. So, yan. So, dito kay additional scenarios is sila naman yung nag-undergo kung saan very applicable din na ma-identify kung ano yung microbes na tutukoyin or tinutukoy nila or tutukoyin din natin sooner or later. Ayan, isang halimbawa ng criminal investigation and microbial forensics is yung pag-investigate kung ano yung mismong halimbawa kinamatayan ng isang tao, kung pwede poisonous ba yung nangyari or infectious disease ba yung nangyari. So yun, that's one of the examples. So all in all, we are talking about the application itself ng tamang pag-identify ng microbes, bacteria, or fungi, parasites, and etc. na pwede natin talaga siyang ma-apply in our future. Like this in our healthcare, yan, sa epidemiology, yung pag-analyze sa isang population, kung sino ba yung nakakuha or pwedeng makakuha ng isang disease na nakakahawa. So, ayan, pharmaceutical industry, kung paggagawa ng gamot, kung paano ba um, ma- ma-identify yung isang bacteria, kung paano siya gagawa ng gamot, ganyan, sa additional scenarios. And that's it. And next, okay na pala. So, for our next reporter po is si Jason Saridon. Opo. Thanks. So, microarray-based identification. So, microarray-based microbial identifications relies on the hybridizations of pre-amplified microbial DNA sequences to array species-specific oligonucleotide probes. So, next. Ayan. Microarrays are a versatile tool facilitating the detections and discriminations of different microbial samples on a single slides. So, yun. So, the goal of microarray is to keep the certains that has many portions ng isang body. So, for example, dito is DNA ng stomach lining, DNA ng skin. So, ilalagay mo siya separately. Tapos, ang hahanapin mo doon kung saan siya mag-meet based doon sa DNA na nilagay mo. Yan. So, familiar naman siguro tayo sa transcription and reverse transcription ng DNA. Kasi, kukuha ka dito ng DNA para mag-react sa isa pang DNA. So, next. Yan. Microarray-based platforms typically use similar barcode regions as other PCR-based methods. So, yan. Familiar rin tayo dyan sa PCR-based methods na yan. Ano ba yan? Polymerase chain reactions based dun sa pagkakaalala ko kay Sir Tia na tinuro. So, yan. PCR-based methods is also a way to make many copies of specific DNA region. Kaya siya sinabi na similar barcode. Kaya sinabi na similar barcode ng microarray si PCR-based methods. So, yun. Versatile tool siya because it uses to detect the expressions of thousands of genes at the same time. So, yun. Yung DNA microarrays are microscope slides that are printed with thousands of tiny spots in defined positions. So, yan. That's it. So, yan. For the next topic, Ella Giducos. Thanks.
Good afternoon, sir and classmates. I'm Ella Giducos and I'm one of the presenters of Group 3. So, additional to the other techniques mentioned a while ago, this is the 9th method, which is about fatty acid profiling. So there, fatty acid profiling. It is also known as FAME analysis or the analysis of fatty acid methyl esters. So these fatty acid profiles, they can be used to determine an unidentified bacterial species by matching or comparing it against known profiles. Pagdating sa bacteria, they produce different combination of fatty acids. That's why they are a good target for reliable methods ng pag-differentiate ng bacterial species. Also, yung fatty acid profiling, they're usually performed using the combination of gas chromatography and mass spectrometry. So this is the process of FAME
of FAME analysis. So on the first phase, bacterial culture is grown, and then fatty acids are extracted from the membranes of microorganism, which is then converted to methyl esters. And then the third step, ilalagay na siya sa gas chromatograph to analyze the methyl ester fingerprint. And then the last step is i-identify na yung bacterium. With identifying, it is done by comparing. Like sabi ko nga kanina, it is done by comparing or matching the result with reference chromatograms ng mga known profiles. At saka pala sa FAME analysis ng bacterial identification, yung result ng chromatogram, it is actually unique to each bacterium. So, dyan sa graph, sa bawat peak or sa highest point sa gas chromatogram, nagko-correspond siya to a particular FAME. And yung height nun, it is proportional to sa amount ng cell na present. So, ayan. This is just a quick run-through about gas chromatography and mass spectrometry since I have mentioned them a while ago. So, ayan. Gas chromatography or GC. It is used for separation of interferences, identification of species, or determination of chemical components. And also, it involves gaseous mobile phase. So, ayun, it's a common type of chromatography na ginagamit sa analytical chemistry para for separating and analyzing components of compounds na that can be vaporized without decomposition. So, yun lang. On the next slide is about mass spectrometry naman. Which is also called as mass spectroscopy. So it is an analytic technique by which chemical substances are identified by the sorting of gaseous ions in electric and magnetic fields according to their mass to charge ratios. So ayun, ginagamit siya to accurately measure the mass of different molecules within a sample. And also, it also involves 3 key stages or components. And these are the ion source. Sa ion source, dito yung kung saan yung sample, dumadaan siya sa ionization. Yun. So, yung second one is the mass analyzer. So, dito naman, sinusort out or sineseparate yung ions according to their mass and charge. And then the third one is detector. So it is the part where the separated ions are detected and yung relative abundance is recorded. So yan, that's it for fatty acid profiling. If you want to know more about it, or if you want to read more about our reports, we have provided links at the end of each topics. So you can click them if you want. Ayun lang. Thank you very much for listening. May God bless us all. Thanks.